The process of DNA replication doubles the amount of DNA in each cell to prepare it for the process of mitosis, where each daughter cell inherits a copy of DNA from the parents. Many components are involved in the process, and I will explain them step by step. The first step is initiation. The DNA molecule contains information vital to the replication process. To access the information, the double helix needs to be unwound. While you could do it with high temperatures, higher if the DNA has a higher GC content, the temperature would kill the cell. Instead, the double helix is unwound in a two-step process, which accomplishes the same result in more tolerable conditions. Initiator proteins recognize and bind specific regions of DNA, resulting in the unwinding of nearby DNA, particularly AT-rich regions, forming a bubble with two replication forks. This is important as adenine and thymine have two hydrogen bonds, while guanine and cytosine have three, and would thus require lesser energy to overcome the bonds. Following which, helicase would further unwind the DNA at the replication forks. During the unwinding, the regions of DNA both upstream and downstream of the bubble would twist more. This can be demonstrated by getting a cut piece of twine and opening it in the middle. As such, DNA topoisomerases and gyrases would reflect the coiling. At the same time, the unwound DNA single strands are unstable and will pair with each other or themselves. Single-stranded binding proteins, as their name suggests, would bind to the single strands to stabilize them and prevent the pairing. The next step is synthesis. DNA primases bind to single-stranded DNA and synthesize complementary RNA primers. RNA, which stands for ribose nucleic acid, is similar to DNA except for the sugar group, which is a ribose. DNA polymerase binds to RNA primers and adds nucleotides in the 5' to 3' direction using deoxyribonucleoside triphosphates or DNTPs. While you could technically use DNMPs, deoxyribonucleoside monophosphates, to elongate the daughter strand, considering the structure of the DNA has a backbone consisting of alternating units of phosphate and deoxyribose, only an elongation reaction involving triphosphates is energetically favorable and can occur. Both the process of elongation and unwinding occur concurrently. Due to the unidirectional nature of elongation, the process occurs differently in each strand. On the leading strand, elongation occurs in the same direction as helicase, and thus new information can always be accessed by polymerase. On the lagging strand, elongation occurs in the opposite direction of helicase. Elongation would eventually terminate with no new information available for synthesis. As such, RNA primers are frequently synthesized to replicate newly unwound DNA. This creates small fragments of DNA known as Okazaki fragments. This image shows only one replication fork. A bubble has two. You can see here that each strand of parental DNA has both a lagging and leading strand in a bubble. The final step is termination. The full length of a DNA molecule, known as a chromosome, may have multiple bubbles. Replication stops when the bubbles overlap and reach the end of the chromosome. Remaining RNA primers are removed and the gaps are filled with polymerase with upstream DNA sequences acting as primers, and DNA ligase joins the fragments. The replication process is complete, but with one catch. The chromosome shortens. All the gaps left behind by RNA primers can be filled up, except at the 5' most end, as there is no upstream DNA sequence available for elongation to occur, leading to a shortening of the chromosome every replication cycle. Most of the information loss occurs at telomeres, which are DNA repeats found at the ends of each chromosome and protect vital genetic information. Eventually, telomeres wear out and vital genetic information are lost, resulting in cell death. However, there are specialized cells that possess telomerases, enzymes that extend telomeres and are thus immortal. Additionally, cells that have circular chromosome, such as bacteria, do not have the effect of chromosome shortening as there is upstream DNA available for elongation. You can also observe that each daughter DNA has one strand of DNA belonging to the parents. The replication is thus known to be semi-conservative.